The use of FTA cards, generally for DNA extraction, is a simple procedure and takes a short time in that you can finish your genotyping work from DNA extraction, PCR to gel electrophoresis within a day. This is the procedure for applying and preparing plant samples on FTA cards for DNA analysis. Always wear gloves to avoid contaminating the FTA plant card. Secondly, store all unused FTA cards in a cool and dry place. Thirdly, follow universal protocols. The materials that are required: the FTA plant card, where you extract your DNA, a pestle or a blunt instrument, thirdly, a multi-barrier pouch, also a desiccator. You will need a hard surface underneath the FTA plant card, such as a Harris cutting mat. You need a punch with diameters of 2 or 1.2 millimeters. You need FTA purification reagent to wash the card with, and also isopropanol or TE buffer. You need PCR tubes, you need pipettes, and also pipette tips. You also need a parafilm. Lastly, you also need the plant sample. Label the FTA plant card with the appropriate sample identification. Cover the cutting mat with a parafilm in order to avoid cross-contamination when dealing with many different samples. Take as your leaf material from the plant or on the leaf stored in ice. Place the leaf on top of the FTA matrix with the underside of the leaf facing down. For small many samples, you can subdivide the FTA card into smaller divisions. Overlay the leaf with either parafilm or replace the cover sheet. Pound the sample area with moderate force until sufficient plant material has been transferred to the paper. Verify whether sufficient leaf material has been transferred by checking the underside of the plant card. Use great care not to damage the matrix. Ensure that no large pieces of plant tissue remain on the FTA card, because this will interfere with later processing. So you remove all large plant materials. After 1 hour of air drying, if the sample is to be archived, place it in a multi-barrier pouch and store it in a humidity-controlled, cool, dry environment, such as in a desiccator.